Imagine a vast library filled with the secrets of life. Each book holds a different piece of the puzzle, from the basic building blocks of cells to the complex interactions that make us who we are. This library is the world of Omics, a collection of powerful tools that allow scientists to explore the intricate workings of living organisms at a molecular level. In this journey, we'll delve into 4 major omics fields: genomics, transcriptomics, proteomics, and metabolomics. Each field offers a unique perspective on the complexity of life, providing invaluable insights into health, disease, and the fundamental processes that govern living systems. Prepare to be amazed as we unlock the secrets of life, one omic at a time. Genomics is like reading the instruction manual of life. Imagine having a detailed guide that explains how every part of a living organism is built and functions. This is what genomics offers us. It focuses on the study of an organism's complete set of DNA, known as its genome. The genome is a vast and complex code that contains all the instructions necessary for the growth, development, and maintenance of that organism. This genetic blueprint contains all the information needed to build and maintain an organism. Every cell in our body carries a copy of this blueprint, guiding its functions and activities. Think of DNA as a long, twisted ladder. This ladder is not just a simple structure, it is a marvel of biological engineering. The rungs of this ladder are made up of pairs of chemical building blocks called nucleotides. These nucleotides pair in specific ways, forming the steps of the ladder. The order of these nucleotides forms the genetic code, which determines our traits. This code is like a language, with sequences of nucleotides spelling out the instructions for building proteins, the molecules that perform most of the work in our cells. Genomics helps us understand inherited traits. Why do we have our parents' eyes or hair color? Genomics provides answers by revealing how traits are passed down through generations. It also helps us understand why certain traits appear in some family members but not in others, and how genetic variations contribute to the diversity we see in the world around us. Genomics also plays a crucial role in understanding disease susceptibility. Can we predict our risk of developing certain diseases? Genomics helps identify genetic predispositions, allowing for early interventions and personalized treatment plans. It sheds light on our evolutionary history. How are we related to other species? Genomics unravels the threads of life's tapestry, showing us the connections between different forms of life and how they have evolved over time. Genomics is used in various fields. Its applications are vast and transformative, impacting many aspects of science and medicine. It helps personalize medicine, allowing treatments to be tailored to an individual's genetic makeup. It aids in breeding better crops, enhancing food security and sustainability, and it even helps solve crimes, providing crucial evidence in forensic investigations. The power of genomics is truly remarkable, opening new frontiers in our understanding of life itself. If genomics is the library of life, a vast collection of books containing the instructions for building and maintaining living organisms, then transcriptomics is the symphony orchestra, where each musician represents a different RNA transcript, playing their part in the grand performance of life. It focuses on the study of RNA transcripts, which are like messengers carrying genetic information from DNA to the protein-making machinery of the cell. These RNA molecules are crucial for translating the genetic code into functional proteins, the building blocks of life. Not all genes are active at the same time. Just as an orchestra doesn't play all its instruments at once, cells selectively activate certain genes while keeping others silent, depending on the needs of the moment. Transcriptomics allows scientists to see which genes are turned on or turned off in different cells and under different conditions. This is akin to understanding which sections of the orchestra are playing during a particular movement of a symphony. Imagine a conductor cueing different sections of the orchestra to play, creating a dynamic and ever-changing melody. Similarly, cells respond to various signals by adjusting their gene expression patterns, orchestrating a complex and harmonious response. Transcriptomics helps us understand cellular responses. How do cells react to stress, disease, or medication? By examining the RNA transcripts, scientists can decipher the cellular conversation, revealing how cells communicate and adapt to their environment. Transcriptomics reveals the cellular conversation, shedding light on the intricate signaling pathways and molecular interactions that govern cellular behavior. It also provides insights into development and differentiation. How do cells specialize to form different tissues and organs? By studying the transcriptome, researchers can uncover the genetic choreography that drives the formation of complex biological structures. Transcriptomics unveils the choreography of development, showing how a single fertilized egg can give rise to the diverse array of cell types that make up a multicellular organism. Understanding disease mechanisms is another critical application. What goes wrong at the molecular level in diseases like cancer? Transcriptomics provides crucial clues, helping scientists identify the genetic missteps that lead to disease. By studying the transcriptome, scientists gain valuable insights into the dynamic processes occurring within cells, from normal physiological functions to pathological conditions. This knowledge leads to the development of new diagnostic tools and therapeutic targets for various diseases. By understanding the transcriptomic landscape, researchers can design more effective treatments and interventions, ultimately improving patient outcomes and advancing the field of medicine. Proteins are the workhorses of the cell, carrying out a vast array of tasks from building structures to transporting molecules. These tiny yet mighty molecules are essential for almost every function within a living organism. They are involved in processes such as cell signaling, immune responses, and the cell cycle. Without proteins, cells would not be able to maintain their shape, grow, or reproduce. They are the unsung heroes working tirelessly behind the scenes to keep life going, and catalyzing chemical reactions. Proteomics is the study of all the proteins present in a cell, tissue, or organism at a given time. Imagine a bustling city with proteins as its busy inhabitants. Each protein has a specific job, and together they keep the city running smoothly. Proteomics allows scientists to take a snapshot of this bustling city, identifying the key players and their roles. By understanding the proteome, scientists can gain insights into how cells function in health and disease. This field of study is crucial for developing new diagnostic tools and treatments for various diseases. Proteomics helps us understand, what do all these proteins actually do? Each protein has a unique function, and by studying them, we can learn how they contribute to the overall functioning of the cell. For example, some proteins act as enzymes, speeding up chemical reactions that are necessary for life. Others serve as structural components, giving cells their shape and stability. There are also proteins that transport molecules across cell membranes, ensuring that cells receive the nutrients they need to survive. Proteomics helps connect the dots between genes and their functional products. Genes contain the instructions for making proteins, but it is the proteins themselves that carry out the work. By studying the proteome, scientists can understand how genetic information is translated into functional molecules. This knowledge is essential for understanding how genetic mutations can lead to diseases and for developing targeted therapies. How do proteins work together in complex networks? Proteins rarely act alone. They interact with each other to form intricate networks that regulate cellular processes. These interactions are like the gears in a machine, each one playing a crucial role in keeping the system running smoothly. By mapping these networks, scientists can identify key proteins that are involved in specific pathways and understand how disruptions in these networks can lead to disease. Proteomics maps the intricate web of interactions within the cell. This mapping is akin to creating a detailed blueprint of a complex machine. It allows scientists to see how different proteins interact with each other and how these interactions affect cellular functions. This information is invaluable for understanding the underlying mechanisms of diseases and for identifying potential targets for new therapies. Can we use proteins to diagnose and monitor diseases? The answer is yes. Proteins can serve as biomarkers, which are molecules that indicate the presence or severity of a disease. By identifying specific protein signatures associated with different conditions, scientists can develop diagnostic tests that are more accurate and less invasive than traditional methods. These tests can help doctors diagnose diseases earlier and monitor the effectiveness of treatments. Proteomics identifies protein signatures associated with specific conditions. These signatures can provide valuable information about the state of a patient's health and help guide treatment decisions. For example, certain proteins may be elevated in cancer patients, indicating the presence of a tumor. By monitoring these protein levels, doctors can track the progression of the disease and adjust treatment plans accordingly. Understanding the proteome provides a deeper understanding of cellular processes and aids in the development of new drugs and therapies. By studying the proteome, scientists can identify new drug targets and develop therapies that are more effective and have fewer side effects. This research is paving the way for personalized medicine, where treatments are tailored to the specific needs of individual patients based on their unique protein profiles. The future of medicine lies in our ability to understand and manipulate the proteome, unlocking new possibilities for improving human health. Metabolomics is like eavesdropping on the chemical conversations happening within cells. It focuses on the study of small molecules called metabolites, which are the intermediates and end products of metabolism. Imagine a bustling marketplace with metabolites as the goods being exchanged. Metabolomics allows scientists to analyze this chemical marketplace, revealing the flow of molecules and the overall health of the cellular economy. Metabolomics helps us understand: how do cells break down nutrients and generate energy? Metabolomics traces the intricate pathways of metabolism. How do factors like diet and lifestyle impact our metabolism? Metabolomics provides a personalized snapshot of our metabolic health. Can we use metabolites as early warning signals of disease? Metabolomics identifies metabolic signatures associated with various conditions. By studying the metabolomy, scientists gain insights into the dynamic interplay between genes, environment, and lifestyle, leading to new strategies for disease prevention and personalized healthcare.